음.
이게 뭔가 해야 되나? 오케이 누르나? 엑스도 눌렀는데.
Ah. Okay, hello everyone. I am Kangmin Lee, and I'm going to the general club on this paper named Evolution of Error Correction, Journey for a Speed. So let's start from abstract. Sorry. Okay. Okay, are you done? So how do you think about this abstract? How do you think about it, Sejun?
Thanks. Just
Thank you. For me, this abstract was quite difficult to understand and quite there are many technical terms here. And how about the gap sentence? Is there any gap sentence? Not really explicit. So I would like if I choose one sentence as a gap, I will choose this. The proofreading is typically assumed to be poor when selection for fidelity outweighs cost in energy and the speed of the replications. So this is the problem they want to deal with in this paper. So let's start. Let me start with just very basic biological knowledge. You may be heard about the DNA replication, DNA polymerization. So this is the process of copying DNA. So the DNA consists of these building blocks called nucleotides. So we want to copy this DNA to other DNA copy. To copy that, something called DNA polymerase, which acts as a glue, bind to the site, and then the each nucleotide has its right partner. So that means the A and T should combine, and G and C should combine. So there are four building blocks which consist the DNA. So then C will come to here to bind, make a binding from them. And then the glue will move to the next position and we'll repeat this process to incorporate A, and then they will keep make this kind of binding. And finally, we can successfully copy this DNA. So this is all good. This is very like textbook picture of DNA replication. But in really, sometimes error can happen in this process. Let's consider this situation. There is, in that position, there should not be A, there should be C, but somehow A come here and error copy made. And experimentally, it has been known that the error rate is like decimal, minus 9 power. How do you think about this number, Tegan? Is it too small or okay?
Yeah,
Fine.
because it can never make the error.
Yeah,
If the error rate is zero.
maybe, yeah, lower than that. So actually, this low error rate is very controversial during the 1970s. And let me explain why. Let's compare this correct binding and the wrong binding. You can write this like chemical equations. So the C and A will compete for the position to bind with G. And there is some kinetic constant. And at equilibrium, we can write this kind of Michaelis-Menten type relationship. So we can define this error rate as the rate of the combined concentration, which is just again AC over KW. And the thing is that only with this mechanism, it is really hard to reach this rate of the small rate. So what could be the solution for this problem? The naive answer might be, oh, biologists do experiment wrong and wait for a new experiment, or we can do better. And there's a very good paper in 1974. And you may notice the author of this paper. It is the J.J. Hopfeld, who is the Nobel Prize winner. He's actually known for the artificial intelligence, but he also do this. So when you consider this reaction, he imagined there could be another unstable intermediate between the reactions. With these settings, he proved that the error rate is square of their ratio. And if there is like n steps of these intermediate reactions, the error rate could decrease down to the nth power of that ratio. So now the error rate is kind of attainable with these processes. And really interesting about this paper is that his discussion. He mentioned some of the DNA processes, and he says all these processes are at first sight wasteful, but each is, however, essential side reaction for kinetic proof reading. I think it is very fancy discussion. And so what he did is that he kind of collected these redundant reactions and metabolites, and he integrated this into his own theory. His called kinetic proof reading. So far, any questions?
Everyone is okay about this, yes, it.
How did he like manage to know about the unstable intermediate?
Sorry?
How did he know about the unstable intermediate? Is that just a hypothesis?
Yeah, it was initially his hypothesis. Yeah. But I think he know all these the kind of biology, so he can think of these intermediate reactions.
So like he know there's some idea for in biology. So that intermediate step looks very redundant, right? It looks like waste, but actually it turns out essential. So like an air variable of your car. An air variable of your car looks useless, but when car accident happens, it becomes very, very.
Yeah, exactly. So let me continue. But there are some considerations you should consider with this kinetic proof reading. So first thing is that, so there are more reaction steps. It inevitably slows down these whole reactions. So if you can draw this kind of a simple relation. So x-axis the error rate. So if you want to make the error rate very low, you should use more time to proofread the process. So they have this relationship. And then one more thing is that it requires energy. As I mentioned, they should like create unstable intermediate, which means that they have high energy. So they inevitably use more ATPs to to make the intermediate steps. So the thing is that this process is anyway time and energy consuming. Then why cells do proofreading? Maybe the nature likes accuracy. And so for that, I will start from what does it mean the nature likes something. So I will just very basic about the evolutionary dynamics. So let's consider this example. So once upon a time in Bimec, there was our ancestors. So in order to survive, he made many slides. And this is his some kind of properties, his trait. So you can see that he has very good high lock. And then he has many offsprings. So we are the offsprings. And we found out that there is a mutant happen in these predecessors. And for example, Kyungjun, he can make slide faster. Or I can make the slide more accurate. And if you look at the next generation, so Mina makes one offspring, and Kyungjun 2, I make for next generations. So what happened? So the upgrade mutants succeed in evolution because they make more offsprings here. Then why it happened? We very simply say some evolutionary pressure act on here. So we say in this situation, the nature likes accuracy. So in another term, the accuracy means the higher fitness. So this is what fitness meaning. So this is the whole very, very simple introduction to how natural selection happens. So in one sentence, the genetic trait such as speed or accuracy with higher fitness will survive to go into the next generation. So everyone get it?
There is no Tegan.
No, Tegan is here.
No, he's not Tegan.
Yes, maybe someone else. Okay. Then, okay, based on our previous discussions, this is the picture we have now. We have in one side speed and energy, and other side the accuracy. So the conventional view was that nature developed the error correction because at the cost of speed and energy. It's very natural to speak. And so they would say higher accuracy means higher fitness. So they develop error correction. But if this were a whole story, the title would be changing and it would not be a science paper. They actually turned this view from this view. Actually, what they mean is that at the cost of energy, they would increase both speed and accuracy. But for doing that, we need one more thing, the effect called stalling. So let me explain what is stalling. So here we have again the correct copy and the wrong copy. So here the C is the wrong copy. And we should make the red bone here to extend the DNA string. So it is called the phosphodiester bond. Okay. And it extends the DNA chain. You can calculate formation rate of the correct bond divided by formation rate of the wrong bond. And if you draw this in very different cupping processes, please see the y-axis, the ratio would be 10 to the 5 times slower for mismatched pairs. So if the previous copy is wrong, the next copy will be affected to their process is very delayed. And this is very universal across different copying processes inside this cell. And
So yeah, if card is broken, then so here slower for mismatched pair means what is it slower?
oh,
So so because of to fix it or just
uh,
regular process is slow down.
this bone formation is very slow down.
I see. So it's not except for the fixing, just car becomes slow.
Yeah, just curve becomes slow.
Okay. Error happened, then
Next process is very delayed.
car speed is slow down.
Yeah.
Okay.
So everyone good. And this is something we will call stalling effect of this mismatch pair. So if we consider this path proofreading and stalling, we can change the view from this to this kind of new landscape. So if error rate is too high, then time to copy would increase because the stalling would delay whole the processes. And this is how the view is shifted from this one to this one. So now it's time to formulate the situation with mathematical model, and it will be a long story. So I will introduce this model, kinetic proof reading they use in this paper. So let's look at the state of this site. We can describe this in this manner. So at index 0, this is empty. And if you go to the right side, the right pair will be bound. And if you go left side, the wrong pair will be bound. And each 1, 2, and -1, -2 are intermediate reactions. So here, in this model, two types of reaction is possible. The first one is that you can see the arrows, the transition between the neighboring states. And one more thing is that direct jump and direct jump to the empty state. So 0 to 2 or -3 to 0. Yes. Basically, these two kinds of reactions are possible. And they occur with this rate. So I read this kind of index notation. You can see because they are neighboring ki to I plus 1 and ki to I minus 1. And also we can write one more rate constant, 0 to n and n to 0. Okay, everyone? And if we had this rate, we can write down some chemical master equation to describe this whole reaction network. So what is left is to do, how can we define this rate constant in physically valid sense? So let's do this. Let's first consider this right part, going to the right bond. And first the transition between neighboring states. We will draw this kind of energy landscape picture. So if you consider the two states, 1 and 2, they will each have different energy level in their state. But it is also, there is some kind of energy barrier between them. So if you want their energy level, and if you want to go from 1 to 2, you should overcome this energy barrier. So which we call this energy gap here. So very intuitively, the reaction rate getting slower if the energy gap is bigger. So you can, So you can write this kinetic rate as the exponential to the energy gap. V1 minus V12. And also for the reverse direction, you can just swap the indexes. But in this paper, for the simplest model, they just use the homogeneous constant for their model. So this is simplified to e to the v minus b. Everyone okay? But they consider one more thing. So in this reaction, they consider some kind of driving force that will make the dissociation becomes more easier. So due to that driving force, the reaction to the dissociation direction would be easier. That means the energy barrier is lower, becoming lower. So this rate becomes this rate. Because of the driving force, the reverse direction will become faster. Okay. So the dissociation is faster. Let's consider the second part. And this is the direct jump from and jump to empty state. And to construct the reaction rate, they consider the kind of detailed balance. So what is detailed balance? Let's consider this example. There is some kind of loop of reaction here. Very unintuitive, ACB here. And also there is the inverse reverse direction. So very simple term, detail balance means flux through one direction. The reaction through one direction should be equal to the reverse direction. And you can write this as this multiplication equality. And this kind of guarantees the equilibrium of the whole system. So in our example, you can consider this loop and K1, 0, 1, 1, 2, 2, 0 as same as the 0, 2, 2, 1, and 1, 0. And more generally, we can have this ratio of the NO to OM as the ratio of each neighboring connected rays. So their ratio is also constrained with this equation. So
한번 빼.
네.
A에서 C로 갈 때 A에서 C로 가는 거랑 A에서 B 거쳐 C 가는 거랑
아.
똑같다라는 뜻인가?
A에서 C 갔다가 B로 갔다 다시 A로 돌아오는 거랑 네, B, C, A로 가는 거랑
결국은 똑같아지도록
네.
그러니까 도록 만드시는 대로
네.
스피드가 백으로 돌아가는 스피드가 에너지가 같다는 것이다. 스피드라고 해보자.
네, 스피드.
이거 어려운데 사람들 everyone is okay about this concept?
네.
근데 이걸 가정했어요?
네, 요거는 가져가겠습니다.
혹자는 생물학에는 디테일 밸런스가 없다라고 얘기하는데 에너지가 들어가니까
네.
나중에 나와?
Actually, I didn't really include in this slide, but sorry. Here. If you only assume this part, the detailed balance is okay. However, by introducing this another driving force, actually the whole detailed balance is broken. Yeah. Okay. Okay, let's now consider the part for the wrong direction. Everything is the same, but only the difference is that they allow the dissociation become more easier. So if you again draw this energy diagram, the direction to the dissociation is getting easier, lower energy barrier, and that is the energy gap delta. So because we assume just constant energy barrier constantly, so and this is lower by delta, you can see this relationship. And those rate constant can be written as this formula. So the first one is the same as before, but for the second one, we have the driving force and this eject dissociation. So finally, we have these four kind of parameters, B, B, delta, and phi for describing this whole model. Everybody okay? Okay, then we have this model, and if there is wrong copy, it's bind to here. Let's consider this situation. And as I mentioned earlier, when mismatch happen, then stalling will happen. So they simply wait for the some time before next block starts to bind. So they just add some constant time to this model. So they put tau stall. And you can calculate the whole time to replicate DNA of length n with m errors, which can be right in this equations. And the time replication is sum of time to proofread and the time to wait for the stalling. And actually, the time for proofreading is some kind of random variable following the first passage time process, because they start from the empty state to go to right end or the left end. So this is whole the stochastic time. And with this, we can draw this kind of graph. So x-axis is the error rate. So error rate is the mean of error and divided by total n. And y-axis is the average time of t replication. So you can see this like this curve, like quadratic. So this gives you another landscape for the energy, speed and accuracy relationship. Now we are all ready, and now we can understand the entire what is the evolution of error correction during the first speed. So in simple term, I told you the evolution is the process of genetic trait with higher fitness survive. But if you want to do this in silico, that means the parameters with higher fitness function will survive. So I will give you the algorithm.
Go back.
Yep.
So anyway, if m becomes larger and larger,
Yeah.
because stalling time, I see, becomes longer, but when error rate is lower, to make the error rate is very, very low, we need a lot of
Yeah.
proof. That's why it takes a long time.
Yeah.
So let's see. 땡계를 프로파일하면 네. 근데 저 그래프를 그리려면
네.
to draw this kind of graph, we need to do some calculation. How much we have to tip, what time of the proof time is needed to make a certain and small error. Do we know that relationship?
Yeah, actually they like derive the analytic expression or so with this kind of parameters, they derive the mean of the t-proof rating for do this, and also the average n can be derived with those parameters.
Then what is the difference of the color of this graph? What is the meaning?
This color? This color.
What the gray, black?
They are actually just different parameters. They different driving force. Okay. So, okay. We want to do this kind of in silico evolution experiment. So let me explain the steps. First, we will train these three parameters, related to the energies, B, B, and phi. And we define the fitness function as 1 over power replication, T replication. First, we initialize those values, B, B, and phi, and then compute the replication time for this setting, this parameter. Then we propose the next parameter as some perturbation. So just delta 1, delta 2, delta 3 are sampled from some uniform distributions. And with this new parameter, you compute the time to replicate. And then you can accept this new parameter with probability P, which is minimum of 1 for this exponential the difference of T replication. And PE is just another evolutionary parameter. So this probability means that higher fitness will lead to higher acceptance. So they will evolve to that direction. And you repeat this process: propose, calculate, accept, or reject, repeat. So this is kind of just Monte Carlo Markov chain process. Yeah. So if you do this in silico evolution, you will see as evolutionary time, just repeating step increase, the time to replicate would decrease. It is very natural because time to replicate as a fitness function. And we will use more time to prove it. However, also the error rate would decrease, although we only try to minimize the time. And one more thing is this energy dissipation rate, it increased. So I just put it on the appendix on what is energy dissipation, but theoretically we can define this energy dissipation. So this for plot means this kind of relationship balance. The model pay more energy, this one, for speed and accuracy. So finally, we want to verify if this in silico evolution model is really biologically valid. The strategy is use something called mutagenesis. So what is mutagenesis is that using this wild type like cell, you intentionally do some genetic modifications. And you will have different mutant there. And you can interpret in the reverse direction. Interpret the wild type as an endpoint of evolution. So this can replace the whole natural selection process in a very short time. And we can mimic this in silico. So again, this wild type is replaced by endpoint of in silico evolution study. And we randomly perturb parameter from this to make the mutant. So this is the error rate and time diagram for the simulated in silico evolution. And this one is the experimental evidence of this counterpart. So you can see that for the wild type, they achieve very low error rate and very high speed, which is very similar to the simulated one. Any question? Then, okay, now we have this one. This kind of speed alone can develop the error correction. Then the last question is that can it be like generalizable to different systems, not only this kind of copying DNA, copying RNA? They consider this kind of biological things, microtubules or actin filament. And those are kind of self-assembly of multi-component structures. You can see that they have very like building blocks and they assemble to make these big structures. So they simplify this model as something called kinetic type assembly model. So they are consist of very cute just tiles, and they will gather together to becoming bigger and bigger, and they complete to make some big structure. The thing is that the tiles should be placed in the right order, as very similar to DNA replication. But if the error, some wrong order happens, the binding of the next tiles are becoming slower. So this is very similar to the situation of the stalling. So this is called the kinetic trap. And this is the counterpart of the stalling in this model. And we also need some counterpart of proofreading, which is called the dynamic instability. So what is dynamic instability? You can see the x-axis is the time and the y-axis is dissociation rate. It like periodically increase, decrease, increase, decrease. If this happens, the assembly can like spontaneously melt down to break whole these structures. So this is actually what very similar. And here the correct block can just better survive during this kind of melting down. So this is kind of similar to the kinetic proofreading because they have many intermediate go back and forth. And also here, go back and forth to complete the assembly. And with this model, they also do in silico evolution of this model. And this is very simple. They only tune the parameter, the melting down time. But here, the melting time is this, how long their instability is continued, the period. So as you continue the evolution, because the fitness is 1/t replication, the assembly speed is very fast, and the error rate becomes very slower, as you've seen in the DNA examples. And also, the dynamic instability, that means that just melting down time increase. So again, they interpret this dynamic instability as some kind of energy cost because they disassemble the assembly, so they require more energy to reassemble the structures. So again, the model pay energy for speed and accuracy. Okay, I think this is nearly the end of my slide. So I will give this is the take-home message. So in this paper, they shift the view from the speed energy versus accuracy to energy versus speed and accuracy. And this could be happen by error correction and distorting. And this kind of mechanism could be generalizable across different systems, such as DNA replication or this self-assembly of microstructures. So this is all for my talk, and thanks for listening.
Yeah, please feel free to have a question.
Thanks for your briefing.
네. 아, 네. 네. 아, 요 딥프로프는 얘네가 다리 하나에서 오른쪽 끝까지 가거나 왼쪽 끝까지 가는데 걸리는 시간이면 그래서 총 N개를 만들어야 되니까 이제 각각이 랜덤 배력이라서 N개가 있구나. 여기에선 모델을 더 스폴링 그냥 컨스탄트 타임 더하는 걸 해가지고 만약에 에러가 N번 일어나면은 이제 일단 N 플러스 N 곱하기 타우를 더해줬습니다. 네. 어,
이상한 질문인데 스토리에서 타워는 평소인 거
네.
M은 랜덤 디스트리뷰션 포함.
저 들었어요.
자 N이라는 애 디스트리뷰션.
네.
아 그럼 혹시 저 프로파일 왼쪽에 있는 그러니까 I부터 I1부터 N까지 더하는 건데 I가 3패일까요? 저 I가
I가 이제 각 요거 사이즈들이에요. 그러니까 총 N개 이퀄리티가 저렇게 하니까
네.
그래서 1부터 N까지가 있는 겁니다.
그럼 N 오버 2가 중심인가요? NT인가요?
N over 2.
그러니까 지금 롱 디렉션이랑 라이트 디렉션이 있는데
네.
롱을 첫 번째 I 원으로 보는 건 제가 지금 헷갈렸어요.
요 I는요. 여기 뉴클리어 타입가 N가 아니 4개가 만들어져 있잖아요.
네.
걔 각각이 하나가
I.
네, 맞습니다.
보면은 저 그래프에 따르면 에러가 0인 거는 사실 거의 불가능.
네, 그 걔는 약간 코스타 말이 안 되는 쪽이니까.
I think I understand that part.
Mhm.
How can we calculate energy?
I didn't actually explain, but so because you, oh sorry. So this is kind of from the this is kind of formula from the stochastic model to how to calculate the energy dissipation here. And so they call this the first part as a flux and second part as something force. And this is kind of like 약간 그냥 한글로 할게요. 약간 단위 시간당 한 일이 힘 곱하기 속도가 되는데 그래서 앞에가 약간 속도에 대한 거고 뒤에가 약간 힘에 대한 파트입니다. 그래서 근데 만약에 이제 두 개 딱 평형을 이루면은 제 두 개가 딱 같아져 버리니까 저 더 이상 에너지를 사용하지 않아요. 평형에서는 이제 에너지가 더 이상 쓰지 않는데 이제 만약에 노니키빌리엄이 되면은 쟤네가 이제 0이 안 되니까 에너지를 씁니다. 약간 이게 노니키빌리엄에서 평형으로 가려고 계속 에너지를 방출하는 그런 느낌이 저기 에너지 디시페이션.
디테일 밸런스를 가정을 했는데 거기 추가적으로 드라이브 포스.
네,
그래서 그러면 에너지 디서페이션의 근본적인 소스는 드라이빙 포스인가요?
약간 그렇게 볼 수 있어요. 그래서 드라이빙 포스도 에볼루션 하면서 약간 커지고 그러면 그 에너지 디시페이션도 많아지는.
스톨링이 그거의 핵심 약간 그게 어떻게 보면 에볼루션 할 때 그 중요한 근거가 된다면 스톨링 값에 따른 최적의 에너지 소비 뭐 그런 것도 나오나요?
스톨링 값에 따른 최적의 네네 그거에 대한 약간 이런 U자 그래프 있잖아요. 그거에 대한 최저점에 대해를 어떻게
그래서 최저점이 스톨링 값 하나마다 하나씩
네네 바뀔 수 있는.
스톨링이 0이 아니면 이어 스토피딩을 안 하는 쪽으로 갈 수 있어요.
네, 그렇죠. 스톨링 없으면 사실 아마 프로필딩 안 하는 쪽으로.
근데
여기서 그냥 여기 논문에서 말하는 그거는 실험 데이터가 그거랑 비슷하게 나온 걸로 봐서는 실제 에볼루션이 추구하는 피트니스는 속도다 라고 본 거예요.
그 말을 하기는 되게 조심스럽게 얘기를 했어요. 그냥 하나의 가능성을 제시한 거다. 지금까지는 어큐러시 때문에 생겼다고 생각했는데 사실은 그냥 스피드만으로도 할 수 있을 것 같다 정도로 이렇게 디스커션을 했습니다.
홀링이 일어나는 이유.
저도 잘 정확히 모르는데. So he asked why stalling happened, but they just short-term measure is kind of geometric effect because in the like their protein structure, they have some kind of site there they can very like push away. So, but if the wrong nucleotide here, then they tend to push away. So their bone formation is really becoming slowed down.
Right? So
Sorry?
So to make make DNA strand more.
Oh, yeah, yeah.
Or the DNA polymerase should bind to matter.
Yeah,
If there's a wrong pair,
Yeah.
then maybe the team is part of.
yeah. Okay, any other questions?
Okay, no question? Okay, then, thanks, Kangmin. Yes.